In this short video, I am going to explain about achondroplasia that refers to dwarfism. See, in our population, we find certain persons, actually their frequency is very less, but certain persons are quite short in their stature with some specific deformity. So let us see that how a dwarf individual looks. In this figure, you see this individual is in childhood and the same individual is shown in the adult stage. We observe that they have very large size of head. This is referred as macrocephaly. Enlarged head portion is referred as macrocephaly. And the frontal part of the forehead is actually protruding one. So frontal bossing is seen in them, and portions on either side of nose will be little bit depressed. So this is mid-face hypoplasia. So these are some of the features which you can observe at the head portion. As a whole, you can say that they have large head and their limbs are very short. Okay, so presence of short limbs, short bones of limbs, that is also another important feature. And if you observe the palm of such individuals, they look to be trident. So phenotypically, it's quite easy to distinguish that this individual is a short one. Now, main cause of human dwarfism is actually autosomal dominant disorder. Such individuals have a dominant allele which causes this specific problem, and it is caused by a specific gene mutation. This gene is FGFR3, that is fibroblast growth factor receptor 3 gene. So it is actually a single gene disorder, that is fibroblast growth factor receptor gene, and because of this mutation, one suffers from achondroplasia. So you can say that those individuals who show normal features, they possess actually recessive allele on both chromosomes, and that is why they are normal. And a person showing achondroplasia is actually heterozygous, because this particular gene in homozygous condition will be lethal. Okay, so EFG-EFR3 allele in homozygous condition would be absolutely lethal, and that is why such type of individuals are not found in the population, means at least to their reproductive stage, such individuals will not be there at all. And its incidence in the population is about 1 out of 15,000 to 1 out of 40,000 live births, and it affects all races, means this mutation occurs in almost every race of human being. This trait is useful to analyze rate of mutation in a population. This is quite important point to understand, that since it is a dominant mutation, you can exactly count the number of dwarf individuals in the live births or in the total births, and you can exactly see that out of these many births, if one or two individuals are born with the achondroplasia feature, then what would be the mutation rate? So in a survey of nearly 250,000, that is 250,000 births, the mutation rate, which is indicated by mu, has been calculated, and for this achondroplasia, it has come to be 1.4 into 10 to the power minus 5. So this is the rate of mutation for this particular gene. In this pedigree, you can see the inheritance pattern. As I said, it is autosomal dominant trait. So the allele is actually dominant in nature, and autosomal dominant traits are expressed almost in every generation. So you can see in every generation it is expressed. So this is happening here, because in this particular pedigree chart, you can see three generations. Generation 1, this is generation 2, and then this is third generation. In all the three generations, we are observing the individual suffering from achondroplasia, this black color shows the presence of the trait. So these individuals are actually dwarf. In this case, the male individual is a dwarf one. He gets married to a woman who is having normal trait. Now in the next generation, they have two sons and both are dwarf. So they are achondroplastic persons. And this male individual is married to a woman who is also dwarf woman. So they have five children. See, one, two, three, four, five. And out of these five, three are showing achondroplasia and two daughters, these two are normal. It means the parents are heterozygous. It becomes absolutely clear that this individual is heterozygous, this one is also heterozygous. That is why the recessive allele combine and daughters become normal in their features. In this family also you can see the male individual is dwarf one. This one is a normal female. They have three children, two daughters and a son, and these two daughters suffer from achondroplasia, whereas the son is normal. It means this fellow is heterozygous. So one thing is clear, that homozygosity of this particular allele, that is of achondroplasia, will be lethal. Definitely we can say that here there would be chance of homozygosity, but these three individuals will definitely, if they are live one, they will definitely be heterozygous. So this is pedigree showing inheritance of dwarfism. This is another slide in which we can have idea at molecular level. See here again, three generations of a particular pedigree is shown, and the individuals who suffer from achondroplasia, they are shown in this third generation as well as in earlier first and second generations. And these two parents means they are deceased, they are dead, but their situation there, whether they were dwarf or normal, such things are very well known, and that is why three generations are very clearly mentioned. But what is important to understand, that in this case the restriction fragment length polymorphism analysis has been done, that is RFELP, and in this analysis the purpose is to show autosomal dominant achondroplasia, and wild type allele, wild type means normal allele, is presented by a single band, that is of 510 base pair, and the mutant one, it is shown by two bands, one of 227 nucleotides, or you can say base pair, and another of 283 base pair. So you can say that the mutant allele or its gene segment, DNA segments that gets split by the specific restriction enzyme, and that is why a mutant allele is actually seen in two different bands, that is 227 base pair band and 283 base pair band. So here the third generation individuals, see their DNA content was taken, and that was subjected to RFELP analysis. What we observe here, this shows the markers, okay, the size of DNA bands, and here individual 2, 3, 4, 5, 6, 7, their DNA was actually analyzed for this purpose. We observe that individual 4 is a normal one. So in this case a single band is visible. See this band, whose size is 510 base pair. So its size is exactly 510 base pair. A single band is appearing. It means maternal, paternal, both chromosome carry normal alleles. Whereas if you observe this third individual of the third generation, then in this case you observe one band, it is of 510 base pair, and then two more bands are visible. The upper one is of 283 base pair and the lower one is of 227 base pair. So this individual has three bands. One chromosome carry a normal allele, which is seen in the form of a band 510 base pair, and the mutant one is actually in two different bands, the lower one 227, upper one 283. Here also this fellow is a mutant one, means is suffering from dwarfism. So here also you can see 1, 2, 3 band, one for normal allele and these two for the mutant allele. Again, in case of fifth individual of third generation, we find 1, 2, 3 bands. Okay, one for normal allele and these two is for mutant one, because mutant allele DNA segments get split into two more regions, because restriction enzyme actually recognizes a particular site in that allele. And here this fellow is normal, that is sixth one of the third generation. Here also a single band is visible, other two bands are totally lacking, and this dwarf individual also shows three bands. So it is a very clear kind of a study in which you can see the heterozygosity present in the achondroplastic persons, whereas total normal homozygosity is there in a normal individual. So I think that these are some of the salient aspects regarding the analysis of dwarf individual, and we can have idea that how such individuals show the inheritance pattern of the allele which is actually mutant one to the next generations. And one more thing I would like to add here, in this case, in this particular pedigree, what we are observing that males and females both are showing the trait in almost equal way. Means males are also sufferer, females are also sufferer. So this is actually one of the points to understand that the particular trait is situated on autosome, because autosomal genes or autosomal alleles are inherited in both sexes with equal probability. So we find males suffering from this achondroplasia, females also suffer from achondroplasia, and this shows that, and the trait is being expressed in all the generations. So these are the evidences to say that achondroplasia determining gene is autosomal as well as it is dominant one in nature.